Okay, good morning. So it's Monday, April 20th. This is our biology class, and we're going to continue where we left off on Friday. So Friday, we were looking at mutations, and you guys were asked to post your own original DNA sequence and then change it, right? Meaning mutate it, do something different, do a point mutation, do a frame shift mutation, whatever you want. So I'm going to go ahead and share my screen with you and show you what we came up with. And so I'm going to pull up Schoology for you here. And I'm in the wrong class. Sorry, let's get into biology. So from our biology discussion group, a lot of you posted. Let's look at it here. You guys posted your own mutation. And the question was, what are the effects? Okay, so you guys came up with some different answers. And I'm going to pick up from here. Okay, so what you guys did is you used my example, and in groups last Friday, if you weren't here, what I had you guys do is look at my notes to figure out what's different between these two DNAs, for example, right? And we came up with, and I think it was Noemi that told us that this is different here, and that is a specific type of mutation that we call a point mutation, where we're substituting a letter, okay? And then we came up here, and I believe it was Vivian that told us that the difference here was that we actually inserted something, right? We inserted an extra letter, and then that's what caused this whole thing to be switched so drastically. We're now like the pattern is different, right? Because if you notice, it goes by increments of 3 letters, right? But then by inserting an extra letter, we had this weird little extra T. Okay, and that was called a frame shift mutation insertion. So I looked at the discussion board that you guys put up, and I'm still reading them. I think some people need a little bit more review. So that's what I'm going to do now, because some of these were correct, some of these were a little off. So I'm going to review what mutations are, and specifically what the effects are, okay? Because, for example, I'm looking at Noemi's response, and we said that a protein would change. Well, how do you know that, right? And if some of you already pre-read protein synthesis, which is what we're going to talk about now, then like Noemi, you would know like what specifically the changes are, okay? So what I'm going to do is I'm going to share my screen with you for the effects of these little base changes. So can you guys see my screen okay? Give me thumbs up if you can see my screen. Okay, good. So can you do me a favor? How about Mia? Can you read us the 3 different effects of point mutations?
Okay, so number 1 is silent mutation, and this is change in DNA sequence, but does not change amino acids. Number 2 is missense mutation, and that's the change in DNA sequence that changes protein by changing amino acids. And number 3 is nonsense mutations, and that's the change in DNA sequence that causes a stop.
Mm-hmm. So if you have never heard of these, it's because, for one, it's maybe you haven't read the notes or the chapter, but that's okay, because I'm going to show you how you can figure out what the effect of a point mutation is. And the way I'm going to do that is I'm going to share my screen with you and actually put up my whiteboard. So we want to know if we change something, and again, I'm going to put my DNA here. And again, I'm recording this so you can always rewatch it, okay? So we want to know if we have a DNA and we change it, what happens, okay? So we need to ask ourselves a couple questions. So first of all, what does DNA do roughly in our bodies? If you can give me just a brief summary, what is the point of DNA, anyone? What does DNA do for us?
Replacing damaged ones.
Yeah, thank you, Oscar. So it replaces things, like we said, right? Like if we break a bone, if we need to grow something that was damaged or repair something, DNA's function is to store that information for us as a sequence of letters. So I'll just put some letters here, right? We'll go like A, T, G, C-A-T. And again, all these are just bases, but the bases in particular, they mean something, and what they code for are basically traits. So like Oscar said, if we need to replace like broken bone, these codes of letters are essentially the code for that bone to repair itself. Okay, so I'm going to go a little further than that, because in the discussion board, I wrote my own DNA, and then I asked you guys to change it and try to figure out, well, what actually changes in the protein itself. So we're going to figure that out. But in order to figure that out, we need to be aware of something that I've yet to introduce to you, but I assigned you an assignment on Schoology that was due later this week, but some of you already did it, and that's on protein synthesis, okay? So I'm gonna give you a preview as to what protein synthesis is, and then I'll show you where this comes from in my notes, okay? So I'm gonna just minimize my board, and in order to figure out, like Mia just read us, like what the effects are of these changes, we need to understand what protein synthesis is, okay? And so what I'm going to do is I'm gonna share this image with you, okay? So yes, like Oscar said, DNA makes things for us and it repairs them, but it doesn't do it directly, okay? So this comes from section 8.4 and 8.5. I'm just kind of combining everything. And there is a watch assignment that I sent you on Schoology. It's not due yet, but if you didn't get a start on it, I know Christian was the first one to submit it. DNA, the way that it makes things for us, like Oscar said, is there's these in-between steps where DNA actually has to make something first called an RNA in order for it to make the protein. And the protein is that stuff that Oscar's talking about. The protein is that bone, because yes, bones are made of protein. So if we break a bone, we can't just make the bone directly. We need DNA, which is the instructions, to make this thing called RNA, which will then make for us proteins. The name of that process is called protein synthesis, okay? And that's what I'm going to introduce to you today. And it's something that is broken down into two different steps that we call the transcription and the translation, okay? So first things first, we need to review the cell. Okay, so on this picture here, and we've already looked at cells obviously last semester, but I'm just going to do a quick little review with you. Okay, so what is this big green structure first of all? So this purple thing is the cell. Okay, inside the purple cell we have this green structure. What do you think this green structure is, guys? Anyone? What's this green structure inside my purple cell? Anyone?
I don't see green.
Oh, wait, hold on. You don't see
green.
Oh, you know what? Sorry. I think
Yeah.
I'm sharing the wrong screen with you. I'm sorry, guys. Can you see my screen now? Can you?
Yes.
Oh yeah.
Okay, sorry. I was sharing the wrong screen, my bad. Okay, so to review, DNA, yes, it is the instructions, like Oscar said, to make stuff or repair stuff, but it can't do it directly. DNA needs to make this in-between molecule we call RNA, and what I mean by in-between is that it's kind of like the middleman to get stuff done by the DNA. So DNA will make this structure we call RNA, and then that will make the protein. And the protein is basically all the stuff in our bodies. That's what DNA is responsible for making, but it doesn't do it directly. It needs RNA to make all the stuff we need. So this purple structure is the cell, pretend. The green structure where our DNA is found, what's this green structure called, guys? It's the nucleus, right? So inside the green structure, we have our DNA inside the nucleus. And in the nucleus, we're going to go through this process called transcription, okay, where DNA will basically rewrite itself. To transcribe, guys, means to write, okay, to transcribe. So this process that I label transcription means to rewrite. So DNA will rewrite itself as this structure we call mRNA, okay, mRNA. So it's going to process or, you know, clean itself up a little bit, rewrite itself. And so DNA, in a process known as transcription, will make this new structure called mRNA. And as you can see here, DNA, remember, it's a double helix, right? It has 2 strands. But RNA is different. RNA only has 1 strand. So it needs to rewrite itself and actually change itself. So the DNA will change itself into mRNA. And that mRNA, which again is an RNA, will leave the nucleus. And so if the green is the nucleus, what is all the purple stuff in this cell? What's all this purple stuff, do you think? If the green is the nucleus, what's all the other stuff inside the cell? Think back to your cell analogy poster that you did last semester. What's all this purple stuff?
Cytoplasm?
Thank you. Thank you, April. It's the cytoplasm. So inside the cytoplasm of the cell, we have these little structures we call ribosomes. Does anyone remember what ribosomes do from last semester on your cell analogy project? What does the ribosome do, guys?
Do they make the protein?
The proteins, exactly. So throughout the cell, we have lots of ribosomes. They're everywhere, okay? And the job of the ribosome is to make the protein, right? Because that's what we want to get to. So the name of that process where proteins, like April said, will be made, that process is called translation, okay? So what I'm going to do with you now is I'm going to walk you through the whole process of making a DNA. And here's my DNA again. You guys can see my whiteboard, right?
Yeah.
Okay, so I'm going to walk you through how we get to DNA to RNA to protein. Okay, that's what we're gonna cover today. So DNA, and then we're gonna make RNA, and then we're gonna ultimately get to the protein. And the reason we need to do this is because we need to figure out, well, if we're changing the DNA, like we said last week, what's the effect? And so some of you correctly put, I think it was Noemi that said, like, it changes the amino acid. Well, how does she know that? That's what we're going to figure out, okay? So, spoiler alert, DNA and RNA use different letters. And this is something we talked about briefly last week, but now I really, really want to talk about it. So let me give you first the differences between DNA and RNA, okay? So DNA, if you recall, has a set of rules that we call base pairing rules. Does anyone remember what the base pairing rules are for DNA? Like what pairs up with what?
Adenine to and cytosine to guanine.
Good. So A pairs up with T and G with C, like Alexa said, right? Now, that's for DNA. However, RNA is a different molecule, and it actually doesn't even have a T. It has a U. It has a different letter. So instead of A's pairing with T's, there is a new molecule that we're going to talk about now called uracil. And uracil, we abbreviate it with U. Okay, so wherever you would see a T in a DNA, we're going to replace that with a U. Does that make sense?
Yeah.
Okay. And then wherever we see G, that still pairs up with C. That doesn't change. So the difference is not too big. It's really just a letter, okay? So if I want to figure out what does this DNA make, we need to figure out, well, according to the base pair rules for DNA versus RNA, we're going to have to do a little code switching, okay? So rather than using T's, like I said, we're going to swap them out for U's, okay? So in this example, let's say I give you a DNA that reads ATG, CAT, TAT. If I want to figure out the RNA version of that, guys, we need to figure out, first of all, what's the equivalent, okay? So in a regular DNA base pairing, what would an A match up to?
Thymine.
A thymine, a T, right? However, we're not writing a DNA now. We're going to write an RNA. In particular, we're going to write out a type of RNA that we call the mRNA, right? So we want to know, and again, this happens inside the nucleus, right? All this is in the nucleus, like you saw in the other picture. So if we want to figure out what ATG would be, we're going to use base pairing rules, but rather than putting a thymine, we're going to swap that out for a U, okay? So what would ATG be as an mRNA, guys? Help me out.
UAC.
U, A, C. Did we see how Alexa did that?
Yep.
Yeah, right. It's not too difficult, okay? What would CAT be as an mRNA? How about Vanessa?
GUA.
Okay, so G, U, A. And what would TAT be? How about Vanessa? What would TAT be?
AUA.
Perfect. AUA. Okay. So, sorry for my writing, but you see how it's the same exact thing that we've been doing for DNA. It's the same exact thing you did with your DNA model. However, we're just swapping out one letter. That's all. Okay. So that's called the mRNA. All right. Now, going back to that picture I just showed you, DNA makes the mRNA. All this happens inside the nucleus. Okay. This happens inside the nucleus where a DNA will rewrite itself as this new molecule we call mRNA. All right. But like April told us, that mRNA goes to the structure we call the ribosome, and that's what I'm going to label right here. Now, at the ribosome, we're going to now use that mRNA information to make the protein, okay? And the way we're going to do, oh my gosh, my writing, sorry. And the way we're going to do that is we're going to use this mRNA, but there is a little additional guy I forgot to write down called the tRNA. Okay, and what the tRNA is essentially the mRNA is like the code, right? It's the code from DNA, but well, actually, I should just say this: the M in mRNA stands for message. Okay, it's the message that DNA is saying for our bodies to build. Okay, but it rewrote itself, right? It rewrote itself with the new letters, right? The new U's. The T stands for transfer, meaning if the message says, hey, we need UAC, then we need GUA, then we need AUA. Well, like April told us, these little structures called the ribosome, they're found throughout the cell. So we need to go get the parts throughout the cell, which are essentially the message or the code the DNA is telling us to build, but we need to go get them. And the structure that gets them, guys, is called the tRNA. Okay, so we actually need to rewrite this some more. So if mRNA is saying, hey, we need UAC, we need to rewrite that as a tRNA, meaning we're going to do some more base pairing. So what would UAC match up to? How about Melissa? If mRNA is UAC and we want to find, again, the base pair, but in RNA language, what would that be rewritten as a tRNA, Melissa? What would UAC match up to, in other words? Melissa, are you there?
Yeah. I'm sorry, can you repeat that?
Yeah, so we're looking at this UAC, which is the message that mRNA is giving us, and we want to figure out what would the match to that or the base pair be, Melissa, as a tRNA. So we're talking RNA letters, which are these. So if we're saying UAC, what would the equivalent be down here to UAC?
AUG?
Very good. It'd be AUG. And what would the match to this be? How about Dominic, to GUA? Dominic?
It would be CAU.
Very good. CAU. And how about AUA? What would that be? How about Christian for AUA? Christian, are you there? Might not be there. How about Felipe? What would AUA be equivalent to down here?
AUA.
For AUA, what would the matchup be?
So it'd be, I'm not positive, T,
Would it be a T, guys?
A, what, AUA?
No. Good guess. How
Wait,
about Oscar? What would the matchup to AUA be? To AUA. Oscar, are you there? How about Daisy? What would the matchup to AUA be?
UAU.
Thank you. UAU. Okay. So to review, DNA, which is in the nucleus, rewrites itself as this new message that we call mRNA. So all we did is we rewrote ATG as UAC, CAT as GUA, TAT as AUA. And again, this is the instructions that DNA is saying, hey, repair this bone, for example, right? So this happens inside the nucleus where it rewrites itself. That's called an mRNA. Then out in the cytoplasm, like April told us, specifically at a structure we call the ribosome, that message arrives, okay, the UAC arrives at the ribosome, but it needs something to go fetch those supplies. This is like a supply list. UAC, GUA, AUA, those are like the parts. And in order to assemble those parts, we need to go get those materials. That's what the tRNA is, right? So tRNA gets us AUG, CAU, and UAU, okay? And again, this happens at the ribosome. Now, we have one more step to go. We're now going to see, okay, if that's what DNA wants, how do we then assemble the structure we call the protein? Well, the key is the proteins are actually assembled by these structures we call amino acids. Okay, so, and again, a protein is essentially everything, all the stuff in your body that we need to build. So bones, more cells, hair, eyeballs, hearts, everything, muscle, it's all made of protein. But the building blocks or the little parts of proteins are these structures we call amino acids. So in order for us to build the protein or the amino acids that DNA is requesting us, we have to go through these steps of switching letters up, okay? Now, we want to know, what does all this stuff mean? We want to know what is UAC, what is AUG, what is that, okay? They're actually codes, okay? And so what we're going to do now is I'm going to show you what these codes mean. We want to know, what does UAC mean? What does this mean, okay? And so I'm going to share my screen with you and show you what we call the codon wheel. So I'm not going to erase my whiteboard. I'm actually just going to minimize this. And I'm going to share something else with you. Hold on. So I'm not going to erase that. I'm going to share. Where's my other screen? Okay, hold on. Can you guys make sure I got the right one here? Hold on. Where did I put it? You guys see this, right?
Yes.
Okay. So we want to know, what do all those letters mean? Okay, and the way we figure out what the letters mean is we use this thing called the genetic codon wheel. Okay, and essentially what the codon wheel is it's the way that you decrypt those letters, because those letters actually are equivalent to what we call an amino acid. Okay, so let me enlarge this like a lot. Hold on, because that's too small for us. And again, in class, I would have obviously given you this handout so you can use it, but I'm going to have us, learn it together on this, right? Because this is the technology we have. So if the code that we looked at on the whiteboard, I believe it was UAC, right? That was the first little sequence of letters. If DNA is requesting UAC, the way to figure out what UAC means, guys, is there's a lot of letters here. Notice they go from big to medium to small, Okay, so if the code that I think the first code that we wrote that Melissa told us was UAC, the way we figure out what UAC means is on this wheel, you have, like I said, 3 layers of letters. So the first letter on your code, and I'm going to just write it over here. If the first code is UAC, okay, and we do this in increments of 3, okay? And the reason we do them in 3s, we call that, by the way, a codon. A codon is the 3-letter message that's found on the mRNA. Okay, so if the message that DNA told us to write is UAC, and I'm going to just minimize this a little bit so you can see a little better. I'm going to put it in red, right? So DNA rewrote itself, remember, in the nucleus as this thing we call the mRNA, and they're read in increments of 3 letters at a time. Those 3 letters at a time, we call a codon. That's all, okay? So if the first 3-letter codon that DNA is requesting is UAC, the way you read it is you first find the U. The U is the first letter, right? So we go U and then A. So you just align the U with this A. And the third one is C. So you would go like this. Let me write on your screen. Okay, you would go U, A, C. Do you see that, guys? Give me a thumbs up if you can see what I just did. Okay, good. So UAC, that stands for a specific amino acid that we call tyrosine. When we're writing amino acids, guys, we just typically just write the first 3 letters. So if I say UAC, all you would have to write me is basically TYR. Okay. So this codon wheel is something I'm going to send you, and on Wednesday what we're going to do is we're going to practice rewriting the messages that DNA says that mRNA decodes. Okay, now let me go back to my whiteboard with you. Okay, so on this whiteboard, remember, DNA rewrote itself as an mRNA, and that UAC, you just saw, it actually stands for something. And we said that it stands for the amino acid called TYR. Okay, now check this out, please. Notice I didn't use the letters of the tRNA. The reason is because the message that DNA is requesting is found on the mRNA. The tRNA's job is simply to go play fetch. That's it. Okay, so when you're using that little codon wheel, pay no attention to the tRNAs. Okay, the tRNAs are essentially just like the errand boys that go grab whatever parts the mRNA is requesting. So when we look at codon wheels, only pay attention to the mRNA. So the first message was UAC, and we just wrote that that was the amino acid TYR. The next one is GUA. Okay, so let me go back to my screen and see if you can tell me. So what does GUA code for then? What does GUA code for?
Valley?
And again, I'll just show you how to do that. So you go like this, right? You align the big G. So if we're looking for the codon, which is GUA, right? That's the codon. Codon. Oh my gosh, sorry for my writing. Codon on the mRNA. So we're only looking at mRNA messages. And so we look at the G, which is right here. Then we look at the U, and then we look at the A. So you just align all 3. And you're right, that would be valine, and we would just write VAL. So let me go back to my whiteboard. So you would write here, VAL. What's the next message that we need to decode?
AUA.
Very good, right? We're looking at AUA. Pay no attention to UAU. It's literally just the one that fetches it. Okay, so don't worry about this. We need to look at AUA. So we go back to our code on wheel. And what would AUA code for, guys?
Stop.
What would AUA code for? What would AUA code for?
Isoleucine.
Very good. So you would go A, U.
Isoleucine, or
Yeah.
however you say it.
I know it's a weird one. Sorry, guys. It's pronounced isoleucine, but all we write is the first three letters. Okay. So do you guys see it? AUA. Thumbs up if you saw how we did that. Thumbs up if you saw how we did that.
Yeah.
Okay, good. So let's go back to our whiteboard. Thank you, guys. And now we're going to write ISO. Okay. Now, we went from a DNA to an mRNA to a tRNA to the protein, which is the amino acid. This, believe it or not, Oscar, I know this doesn't look like the damaged bone that you said we need to repair, but that's how our body reads things. It reads it with amino acids because amino acids form proteins, and proteins are literally everything in our body. Okay. So, questions about how I did this, how I figured out DNA to mRNA to tRNA to amino acids. Questions about that?
I get it now.
You guys are good?
Yeah.
Okay, so
Mhm.
here's what I want you guys to do. Okay, so I'm gonna first close this because it'll be part of my recording, but I'm gonna just save it just in case. Okay, so I'm gonna close my whiteboard. And what I'm going to do is I'm going to send you guys a code on wheel. Like I said, in class, my plan was to give you one of these wheels and let you guys kind of decode those messages for yourself. Okay. Obviously, we can't do that. So what we're going to do is on Wednesday, I'm going to give you a series of DNAs, and then I'm going to ask you to write the entire sequence of mRNA, tRNA, amino acids like we just did. However, like I think it was Noemi was able to figure out, if you change something though, you technically, like Mia told us, can change the amino acid in three ways, and they were called missense mutation, nonsense mutation, and let me share that slide with you again. And it was the slide that Mia read us. It was right here, right? It can occur in one of three ways. So in order to figure out, well, did it really change the amino acid or change anything at all? You had to first learn how to write the entire sequence out. So now that we've written out an entire sequence, let me review what Mia told us at the beginning, okay? So a silent mutation is a mutation where, yes, you change something, you change the letters, but it didn't change anything at all. Even though you swap something out, nothing changes whatsoever. And in order to explain that, let me do one of your examples from your whiteboard discussion. Okay, so I'm going to put one of the ones that you guys did, and I'll just use here. Let's just do Christian's because I have it up right here. Okay, so we're going to look at Christian's mutation, and we're going to figure out if that truly would change the amino acid. Okay, so check this out. You guys are going to help me. Okay, today's just review. So I'm going to do, how do I erase all this? Okay, good. So I'm going to put up Christian's, and Christian's, your sequence. Oops, what did I do? Christian's sequence was A, C, G, T, A, C. I'm just writing it here, guys. Sorry. G, C, A. Oh, that's a C. G, C, A. T, G, T. And then Christian changed it to A, C, G, T, A, C, G, G, A, and T, G, T. So forgive my writing, but what did Christian change on the DNA, first of all? What did Christian change?
In the third set of bases, he changed the C into a G.
Very good, right? He changed right here, as you can see, he changed that C into a G. So now we want to know, and I'm going to ignore the other letters. We're only going to look at these three sets, okay? So Christian changed the C to a G, like Alexa said. So we want to know, well, what does that actually do? What's the effect of that? And so remember, this is a DNA, okay? So in order to figure out what actually changed, guys, we need to run through the same exact steps. We need to figure out what's the mRNA, what's the tRNA, and what amino acid does that code for? Okay, that's what we want to figure out. So help me out here, okay? So if the original was GCA and he changed it to GGA, we need to first figure out, well, what does GCA actually code for? Okay, so only looking at GCA, okay? What is the mRNA for GCA, guys?
CCU.
Very good. CCU. What would the tRNA for that be?
GGA.
GGA. And now we want to know, to figure out the amino acid, am I going to use the mRNA or the tRNA?
mRNA.
mRNA, right? So we're going to look at CCU. Okay, so let me go back to my code on wheel for you. So we're looking at CCU. And the wrong one, sorry. Okay, so what amino acid does CCU code for?
Proline.
Say it, everyone.
Proline.
Proline, right? So you can see right here, CCU is proline. So let's go back to our whiteboard and we're going to put CCU is proline, right? So we put pro, okay? So that's the original, okay? Proline. But what Christian changed was one letter, right? We changed the one letter. And we want to know, does it change proline though? Will it be a different amino acid? So let's find out, okay? So the change that Christian made, and I'm going to do it in a different color, the change that Christian made for us was, instead of GCA, it's GGA. So now let's look at GGA. What mRNA would that code for, guys? What mRNA would GGA code for? What mRNA would GGA code for?
I think
It's
we
the
accidentally did the mutated one.
Oh, I did the mutated, my bad. Okay, sorry, the original now, I'm so sorry. So let's look at GCA. What would GCA code for?
CGU.
CGU, and I'm putting it on top because that's the original, okay? And what tRNA would CGU code for? What tRNA would CGU code for?
GGA.
Say it louder.
GGA.
For CGU, we're looking at CGU.
Oh,
GCA.
Thank you. GCA. So now we want to figure out, are we going to use the G, or are we going to use CGU or GGA when we look at our wheel?
CGU.
CGU, right? So we want to know, does CGU code for something else besides proline? So let's find out. CGU. So we go back to our wheel. What does CGU code for then, guys?
Arginine.
So,
arginine.
right, so CGU is something called arginine, right? So let's go back to our whiteboard. So now it codes for ARG. Is that the same as PRO, proline?
No.
It's not, right? It's not. So obviously, yes, it did change. So let's go back now to our original post that Christian put up. So you're absolutely right that it was a point mutation. Okay, I'm just going to use no emus here. It was a point mutation, and like you said, it was a swap between a G and a C. So the question is, did that change the amino acid? Yes or no?
Yes.
Yes.
It did, right? It went from what to what? Let's go back. It went from what to what?
Arginine to
Yeah, it changed from an arginine to proline, right? It's a completely different amino acid. But like Mia told us, there are 3 different mutation types that we can get here, okay? And the 3 different mutation types are called silent, means that nothing changed at all. So was that a silent mutation? Did we not change the amino acid?
no.
Was it a silent mutation?
No.
No, right? Because a silent mutation means that we do not change the amino acid, but the amino acid did change. So it was not that. The other type of mutation, like Mia told us, is called a missense, where we are changing the amino acid. So did we change the amino acid?
Yes.
We did.
Yes.
We did, right? The third type is called a nonsense mutation. Nonsense just means, like Mia told us, that it causes it to stop early. And what I mean by stop early, if we look at our wheel, again, where's my wheel? If you look at the wheel, there's actually, you see where it says stop right here? You see how it says stop? It says right here, like it says UGA stop. You guys see that? Okay. There's also one called UAA stop. And do you guys see a third one on here? Do you see another stop on here?
UAG.
Very good. There's a third one called UAG. So meaning, if not only do we change it, like you just saw, but if you cause it to stop earlier than it was supposed to, then it is called a nonsense. So did we change it to a stop? Did it turn into a stop?
No.
No.
No, right? No. All we did was we swapped it from proline to arginine. That's all we did. So let me put up the mutation types. So was it a nonsense mutation then? Did we cause it to stop?
No.
Did we not change the amino acid?
No.
So which mutation type was it? Silent, missense, or nonsense?
Missing.
It's called a missense mutation. Okay. I know that's a lot. I know that this section has a lot of terminology, so don't worry. We're going to go very, very slow. Okay. What we're going to do on Wednesday is I'm going to give you guys essentially DNA strands that read, the letters like we've been doing. Your job is then going to figure out what's the mRNA, what's the tRNA, what's the amino acid, and what changed. Okay, so today you demonstrated that you're getting it, but obviously there's still more to talk about. So what I'm going to introduce you to today, and I apologize for this being a lot of content, but it's a lot of important content. Okay, so just to review with you, we're looking at section 8.4, 8.5, and 8.6, but I'm going to combine 8.4 and 8.5 on one. Next week, we'll be looking at 8.6, and for 8.6, you're going to be reading an article on Newsella, which I'll talk about a little later, not this week. If you got a head start on it, fine, but don't worry about this yet. We're only looking at 8.4 and 8.5. And to summarize, this is 8.4 and 8.5, okay, where we're going to look at how DNA turns into an RNA, which turns into proteins so that we can make all the stuff our bodies need to. Now, obviously, this isn't a very, very short process, right? Because there's a lot of notes that we're going to review. But if you could do me the favor of previewing the rest of my notes here, and I'm going to just cover the basics with you and then let you practice those on Wednesday, okay? So first of all, the name of this process, like I said, is called protein synthesis, but its other name in genetics is called the central dogma of genetics. And dogma just means like the rules, okay? So essentially protein synthesis is just the rule of genetics. And the rule of genetics is that in order for us to make a structure like a broken bone or something, like Oscar has said, right? In order for us to make stuff or repair stuff, DNA has to turn into the RNAs you just saw in order for it to make proteins, okay? And that happens in this sequence of steps, okay? Where the DNA inside the nucleus rewrites itself as an mRNA, exits the nucleus into the cytoplasm, like April told us, and at the ribosome, it makes those proteins as these little balls. And these little balls, guys, these are the amino acids. So essentially, this picture, called the central dogma of genetics, just summarizes the entire process, okay? So what I'm going to do is I'm going to just end right here, because I know this was like a lot of content, but just know that for Wednesday, I'm going to ask that you're able to go through these steps. You're able to, from a DNA, figure out in transcription what is the RNA that was made, and then what gets translated into a protein. Does that make sense, guys?
Yes.
Okay, okay. So I'm going to end here. As always, I recorded this. Please, please, please don't worry about like the immense amount of notes for this section, because I'm not collecting notebooks. Remember, I'm not asking you to copy any of this down. I'm asking you to apply it. All right, so Wednesday, what you're going to do to apply it is I'm going to give you, like I said, I'm going to give you some DNA codes. You're going to rewrite them, and then you're going to use that codon wheel. So I'm going to give you guys a copy of this. And if you have an iPhone, oh, actually, you know what? I'll just put it on your iPad. There's actually an app, and let me see if I can show it to you, because I just put it on my phone. There's an app for this genetic stuff. Let me see if I can get it. Hold on. I'm going to try to share my phone screen with you so you can see it. Let's see. Let's see. Okay. Share screen. Okay. You should be able to see my phone in a little bit. Okay. Can you guys see my phone? Can you guys see my phone okay?
Yes.
Okay. So check it out. There's actually an app for this that I'm going to add onto your iPad where, you know, rather than using this wheel, check this out. If I want to see what, for example, GCA codes for, you see how it lets you kind of move these little wheels around to write your codon code. And it shows you right here the name of the amino acid. Now, this is a little more advanced. This is actually this is a college app where it actually shows you the chemical structure of the amino acid. I'm not going to ask you to worry about this, but I am going to ask you to use this as a way to quickly decode the name of the amino acid. Okay, so that's what we'll do on Wednesday. I'll push out this app to your iPads. If you don't have the iPad, that's fine. I'll just give you the paper copy like this so that you can figure out the codons in Zoom groups. Sound like a plan?
Yes.
Okay, okay. Any other questions other than the mutation and the protein synthesis stuff? Does anyone have any questions about the assignments we're working on? So as of now, we've finished the DNA stuff with your discussion board. So I'm going to continue reading your DNA codes that you guys put up. And again, that's the ones that we just walked through, right? It's these. So I'm gonna look at all these. I'm gonna give you feedback. Some of them, like I said, were very good. Some of you, I think, need a little bit more review. But do me a favor. If you didn't put, like Noemi put, that it changes the amino acid or not, I need you to revise your answer and tell me, it's a nonsense, it's a missense, or it's a silent mutation. Okay, now that you've done that, and so if you haven't put an answer up at all, you can go back and add this, because again, this is not a test. This is a way for you to practice. So please, you can go back and revise your answer, and you can tell me that it changed the amino acid, it didn't change the amino acid, or whatever. Okay, so I'll give you some time to do that while I'm grading them, and I'll give you feedback, and please just change your answers if you need to. Okay, so I'll go ahead and end class there. If you don't change your answers, don't worry. It was just meant to be practice, okay? But we are going to have a quiz on this next week. So you obviously need a lot more practice for how to decode DNAs and figure out if they were mutated, all right? So any other questions about the mutation stuff or the protein stuff today? So thumbs up if you're good and you understand what we're going to do on Wednesday. Okay, okay. All right, guys. Thank you so much. I know that that was a lot today, but as always, you can review this and check out my YouTube link for this and reach out to me. Remember, I do my office hours every Tuesday, Thursday. So if you need more help or more review on this, please message me today or tomorrow. Okay. Other than that, peace out, guys. I'll see you on Wednesday. Bye, guys.
Bye.
See you.
There was a
Bye.